Good afternoon, fans of publicly supported science, and welcome. I'm David Gilbert from the U.S. Department of Energy Joint Genome Institute, or JGI, coming to you live today from Lawrence Berkeley National Laboratory, also known as Berkeley Lab, in the beautiful hills overlooking San Francisco Bay and the Golden Gate. It's where we bring science solutions to the world. We're gathered here this afternoon to be regaled with concise science stories from early career researchers soon to be housed in what will be the newest building at Berkeley Lab. To help me introduce these presenters, I've recruited talent emblematic of Berkeley Lab early career researchers. You know her as the champion of the 2021 Berkeley Lab Early Career Research Slam, and she went on to capture the crown of the inaugural Bay Area Research Slam, where she rose above a field of luminaries from Lawrence Livermore, Sandia, and Slack National Laboratories. She charmed us all with a tale of microbes from fish guts that convert seaweed into sustainable biofuels. I present to you now from the Joint BioEnergy Institute, JBay, the champion, Lydia Rachbauer.
Thanks, David. Your words are flattering. I'm honored to be here today. Thank you. Today, we're bringing you lightning presentations by some of the leaders of research now distributed across the vast Berkeley Lab campus that will soon be brought together in BioEPIC, it's a hard name, so for me. So BioEPIC, the Biological and Environmental Program Integration Center, when the building is completed in 2024. This 73,000 square foot BioEPIC building will showcase the research you will soon hear about by scientists from the Biosciences area, or BSA, and the Earth and Environmental Sciences area, EESA, who are supported by the Biological and Environmental Research Program, BER, of the DOE's Office of Science. I hope you got all of these acronyms because there will be a test later. Seriously, not kidding. It is a lot of information, yes, but I sense you're all up for the challenge.
I am. Are you, Lydia? Fortunately, we don't have to be too far up to the challenge. It's you, the audience. Now, here's what you really need to know about the BioEPIC Slam. You'll hear from 4 leaders of scientific focus areas, or SFAs. They, in turn, will introduce 2 researchers from each of these SFAs who will seek to encapsulate complex science stories into a compressed 3-minute format, moreover, in understandable language. But of course, you, the audience, will be the judge of that, because you will have the opportunity to cast your vote for the favorite BioEPIC slammer. And through the magic of computing science, the honorable mention, second runner-up, and the first ever champion of the BioEPIC Slam will be identified. I'm very excited. We assure you it'll be truly epic, BioEPIC, in fact. Moreover, these presenters will not only be honored with your praise via comments that you put in the chat, but they will be awarded cool cash too. Not crypto, real US greenbacks. Regardless of all these science storytellers you'll soon hear are winners in their own right. Don't you agree, Lydia?
Absolutely. So, okay, let's get started with some trivia. Here is the first question, and it should be an easy one if you paid attention a minute ago. So, what does BioEPIC stand for? Is it Biology Excellence Put into Commission? Is it Biological and Environmental Program Integration Center? Or Biological Exploration and Persuasion in Congress? Or is it nothing? It's just EPIC. Let's see. Just a couple more seconds left.
Clock is ticking. Let's hear it. Were you paying attention?
Everybody paid attention. Of course they did. We have the best audience ever.
Bravo.
Of course, you know it's Biological and Environmental Program Integration Center. Rolls right off the tongue, my tongue at least. So David, why don't you tell us about who's involved in BioEPIC?
Indeed, I will. Thanks, Lydia. Let's meet the 4 BioEPIC anchor tenant scientific focus area leaders who will in turn introduce our SLAM contestants. We start off with the estimable Adam Arkin, a man of many hats. Adam has one proverbial foot on the hill at Berkeley Lab as senior faculty scientist in the Environmental Genomics and Systems Biology Division. He is Chief Executive Officer and Chief Scientific Officer for the DOE Systems Biology Knowledge Base, otherwise known as KBase. That other considerable footprint of Adam's is planted securely on the UC Berkeley campus in his role as a professor in the Department of Bioengineering. And aside from his formal professional responsibilities, you'll be glad to know that Adam is a foster father to many a dog in need. So without further ado, but for today's event, Adam dons his hat as principal investigator for ENIGMA, a consortium of investigators across the country, led by Berkeley Lab. The focus of ENIGMA is to create a predictive understanding of how microbial communities influence their complex ecosystems. Perhaps he'll even tell us what ENIGMA stands for. Adam?
Hello, and thanks, David. Let me share my screen here. Get this one thing right, everything will be fine and fantastic. How is that working for y'all? Good?
That's perfect.
Yeah, excellent. So, David, I am not going to tell you what ENIGMA stands for.
It'll be a mystery.
It is. We've all forgotten it because it doesn't matter. What matters is that we are after a particular grand challenge in microbial ecology. So let me explain. You guys know, you can't have avoided it, that we are surrounded by microbes. We've heard the term microbiome. They're in us, they're on us, they're around us everywhere. We're certainly been affected by microbes over the past few years, as you know. This extends below us into the earth, and in the shallow subsurface, they perform amazing services for us in the sequestration of carbon, the mobilization of phosphorus, the decay and weathering of soils and sediments, the support of plant life, and so on. What ENIGMA is on about is specifically trying to understand molecule per molecule, organism per organism, gene per gene, how organisms figure out where they should live, how they should talk to each other, and how they can perform the jobs that they do, so that we can predict what will happen as, for example, our climate changes, or we can intervene to prevent degradation of our environment. Now, ENIGMA in specific is interested in places where, for various reasons, we have degraded the subsurface by some form of anthropogenic intervention. The graph I'm showing you right over here is a map of the United States, which is colored by places where there is an overabundance of nitrate in blue or uranium in red. This is a USGS plot right here. And these regions that you see are places where there's too much of it. It's not good for us. And it's mostly in things like, here's California where we are. This is the Central Valley where we've over-fertilized the agricultural soils. This is the Ulala Aquifer, which is where a major catchment for water from which we pump the water to water our crops in the breadbasket. And we've overpumped it, and we have over-fertilized, and we've created a dissolved mineral pool of uranium and nitrate that is not very good for us as well. And actually, it's a fairly critical problem for us in the world. But we don't work in either one of these areas. We work in this little dot over there. But that little dot has scale. This is the Y12 site at Oak Ridge National Lab. It's in a classified location called Y12. You can see how high security is because we have a photograph of it. And the point of this is that we work out here. We dig down into the earth near places where we've dumped a lot of radioactive materials. This is very high uranium, very high nitrate. We measure what goes on in this environment down to the point where we understand the three-dimensional shape of the earth and its structure. And we drill observatories into the earth, and then we measure all sorts of things, the chemistry, the hydrology, the organisms that are present, when and where. This is depth along this column here, which organisms are present and which genes are active. And what we're going to show you today is how the people who start, the whole team working on this part, the people here today are people taking this is starting material, isolate the organisms, understand them in the laboratories. So with that, I'm going to hand you over to two of our great young researchers in the project who are in that bag. Once we know who's there and what they have in them, what are they actually doing? So to help you with that, I'm going to introduce you. I think I should introduce you next, right, David?
That is correct, sir.
I'm going to go ahead and stop sharing my screen. I think I can somehow. And I'm going to introduce the first of these researchers, who is Feng Chao-Soon. He's a postdoctoral researcher with me. He's been with me for about 5 years. He's an amazing researcher. His talk is from Fast and Furious, unlocking the next generation of experimentation, really trying to understand how these microbes operate in the world. So Feng Chao, take it away.
Thank you. I'm on the screen now? Cool. Since the first modern computer, ENIAC, was invented in 1940s, computers have become much faster and smaller. The smartphone today is way more powerful than the 1,800 square feet ENIAC, not to mention the supercomputers at Berkeley lab. The improvement of computational capacity both sent people to the moon and allowed me to talk to you virtually now. In contrast to computation, experiment remains pretty much like ENIAC. The current high throughput experimentation is based on the 96-watt plate, which is a big boost from using a single tube by doing 96 in parallel. But is it enough? To understand the function of microbial communities, it typically requires measuring thousands of microbial growth and millions of microbial interactions. Using 96-watt plate, it will consume hundreds of thousands of plastic plates and tons of plastic consumables and reagents, which will cost millions of dollars and put huge burden on the environment. But they are not the worst part. If you are a graduate student doing this research, you will need at least 25 years to graduate. Could we make the experimentation faster and smaller like computation? My research in ENIGMA is to develop the next generation experimentation using droplet microfluidics. By designing the chip as small as a credit card, I can generate millions of water in oil droplets in 1 hour. Each droplet performs like a well in 96-watt plate. Then I use genetic marker to label each drop and measure the growth of microbes in millions of drops parallelly in days, but not years. Moreover, because the droplet is at the volume of picoliter, which is 10 to minus 9 milliliter. So this experiment only costs a few test tubes and milliliters of reagent. I have demonstrated this method by measuring hundreds of microbes under 100 conditions in 2 days, which extended our ability to study the complex microbial community. More than this, we can also use this method to accelerate many other pipelines, including screening novel antibiotics, discovering new gene functions, and detecting the most efficient enzyme. Thank you.
Thanks, Feng Chao. That was awesome. Thank you. Our next speaker is Li Wen, we know it as Audrey Wang. She is a research associate who's been with the lab for 8 months, and she's going to talk about deciphering microbial languages in the contaminated aquifer. So Audrey, please take it away.
Hi. Okay, is my screen on? Yeah. Yeah, so as we know that the ENIGMA site, the Oak Ridge aquifer, it's highly contaminated with high concentrations of heavy metals and uraniums, and also high concentration of nitrates and low pH. So our questions will be, what are the microbial composition and dominant species at this site? And to answer these questions, we need to do high enrichment, high throughput enrichment, as well as sequencing to understand what's the dominant one in our contaminant site. And the second question we have is, what are the major selected stressors that determine this microbial composition? And the final questions that we will have is, which genes are responsible for the survival of these dominant bacteria? So by doing so, in the lab setting, we usually use, just as Fungal said, the 96 well plates to try to simulate the groundwater conditions and composition into all these small well plates, so as we can see that whether the OD growth of different bacteria who will be the dominant ones in those wells. So you can see that here there's a Venn diagram, and that's showing like different composition of toxic inorganic ions that can favor the growth of certain bacteria and suppress the growth of the background bacteria. So here, the middle ones, we identify the major selected stressors, the major selected metals that are considered as the important ones that can favor the growth of the bacteria we're interested in. And eventually, we will use the genetic approaches to study what are the genes that are specifically important for the survival of these bacteria under such circumstances. So in the lab, we usually use transposon sequencing to kind of disturb the genes in the bacteria and to see whether the bacteria eventually grow or dead, and to see, in this case, we can know that what's the importance of these genes to this bacteria. So we'll continue using this approach and extend our scouts to study different bacteria that we're interested in the Oak Ridge aquifer.
Thanks, Lydia. That was awesome. Now hand it over to Bill.
That was really great, Fancheng and Audrey. Are we ready for the next trivia question? I sure am. All right, put on your thinking caps. The question is, when is the BioEPIC building slated to open its doors? Is it 2022? Is it 2030? Is it 2052 or 2024? Your answer, please.
That's such a hard one, David. I don't know.
I think we gave it away earlier if people were paying attention.
Chris, we want to engage our audience.
We'll see. Is the audience engaged? And the answer is, ooh, they were indeed.
Pretty good to me.
That was very impressive, I have to say. So Lydia, take it away.
All right, now we'll hear from ESA Senior Scientist Bill Riley, the leader of Belowground Biogeochemistry SFA, where they conduct basic research on terrestrial biogeochemistry with a focus on soil carbon cycling. Bill and his colleagues seek to answer the question, what will be the role of soils in terrestrial feedbacks to atmospheric and climatic changes? You should know that Bill is an avid guitar player who we have heard is a disciple of Jimi Hendrix and Andres Segovia, depending on the situation. But instead of shredding his axe, we have asked Bill to channel his talents into describing his SFA. Bill, take it away, please.
Thanks, Lydia. I'll say more a Leo Kottke fan than a Segovia fan, but let me get my screen here. Okay, and the other thing I should say is that Margaret Torn is the PI. I'm a scientist on the project. And we work, if I can make my screen go here, oops, sorry. In this SFA to develop an understanding of processes and also to develop mechanistic models of soil biogeochemistry and how they interact with climate. And our particular interest, of course, is that the terrestrial biosphere is an important player in the overall response of the atmosphere and warming to anthropogenic change. And we do this in several ways. We do experimental manipulations, we perform observations outside of those manipulations, and we also do numerical modeling. So I'll give you a brief description of each of these components. With respect to the manipulations, we have two primary sites. One of them is in the Sierras, the Blodgett Experimental Forest, which is a UC facility. There we've been warming soils, so and I'll show you a picture in a second, below ground for about 8 years. We also now have a site established at Point Reyes near to Berkeley. Those sites, the warming sites have been set up, and the warming should start at the end of this year. Let me show you a picture of how, I don't know why my thing is not working, of how the manipulation is done, just briefly. You can see, this is at Blodgett Forest, you can see the ring of conduit, and then there are rods going into the soil about 1 meter that are heated with, there's a control system compared to the adjacent control plot. We can manipulate, for example, to a 4-degree warming, and then we make a suite of measurements on the surface for surface gas fluxes, microbial measurements in the soils, abiotics or geochemical measurements as well, that will be described in a second. We are interested, of course, in understanding the mechanisms responsible for the overall system response to warming, and we break that up into several components. We're interested in the plant responses, because even though we're focusing on the soil most of the time, the original source of that carbon is from plants, and they affect everything about the decomposition. We have a lot of work ongoing on microbial processes, the microbial physiology, their response to stresses, etc. And then we also have a component of the project working on geochemistry, and Mike will discuss some of that work in a moment. And as I mentioned, our ultimate goal, well, one of our important goals is to develop models that can be applied across scale. So we work on site level models up to global scale models, integrating what we're learning at our sites, with the ultimate goal here of predicting at the global scale carbon cycle processes that interact with climate change. So that's a brief description of the project. And now I guess I should introduce Mike Rowley, who is a Swiss National Science Foundation postdoctoral fellow. He's working in our group, and he'll give a little talk now on the effect of calcium on carbon cycling in the soils at our Point Reyes site. Take it away, Mike.
Thanks, Bill. Hi, everyone. Today I'm here to talk to you about soil types, and more specifically, what we've been doing at Point Reyes, California, just located to the north of San Francisco Bay. I'm a soil biogeochemist, and what does that mean? Well, as you can see from the inverse diagram up here, the soil up here, soils are a matrix of organic material mixed with rocks or minerals that different liquids and gases pass through. As a soil biogeochemist, I look at how these different components interact through the chemistry. One of the major elements that I'm interested in, as well as the TSSFA, is carbon. We're interested in this due to the pressures that are being placed upon global systems by human populations. What most people don't know, though, is that soils are the largest terrestrial carbon reservoir in permanent exchange with the atmosphere. Much like us, they constantly respire CO2, and they do this as microorganisms break down the different organic components of the soil. So much so that soils naturally emit about 2 times as much CO2 as humans do through fossil fuels every year. But the big question is, how will this soil carbon respond to future change, such as warming, and how is this carbon protected in soils? Well, most of my research is focused on how calcium, an important macronutrient for plants, contributes to the protection of this soil organic matter. It is now established globally that calcium can contribute to the preservation of organic matter in different soils, but this is where they typically contain calcium carbonate or lime, like the White Cliffs of Dover from the UK, where I'm from, or the Alps. This is unlike the acidic coastal grassland soils of Point Reyes, where we don't always have carbonates. So to establish if calcium is influencing soil organic matter and carbon at Point Reyes, we use a powerful particle accelerator called a synchrotron that you can see up here at the Advanced Light Source on the hill at Berkeley. This particle accelerator creates an extremely strong source of X-rays, which we can then crash into our soil samples taken from Point Reyes, as you can see around my shoulders, and gain new insights into the carbon cycle. These X-rays have so far demonstrated that on a very small scale, contrary to the current belief in soil science, calcium is indeed associated with soil organic carbon, particularly carbon that has a more plant-like signature, even up to a depth of 70 centimeters, suggesting that it's contributing to the preservation of organic matter at Point Reyes. Now what we want to do is we want to build on this early work and investigate how this carbon associated with calcium will respond to future change, such as warming, as part of the TES-SFA. Thanks for listening. Cheers.
Thanks, Dr. Rowley. Next, we have Jing Tao, who has been working in our group for a couple of years and is now working on our scientific focus area. She's going to speak about the modeling component of the project and understanding the response of global soil carbon dynamics to warming. Go ahead, Jing.
Thanks, Bill. So as Mike mentioned, soil contains much more carbon than the atmosphere and play a critical role in climate change. Soil organic carbon can be decomposed by microbes and released into the atmosphere as carbon dioxide. We know it's a greenhouse gas. Or it might be absorbed by mineral surface and stored on the ground for a long time. We know soils are expected to get warmer and warmer over coming decades, and this will increase the microbial decomposition of the soil carbon, soil organic carbon, and might release more CO2 from the soil into the atmosphere. Meanwhile, the increase in microbial activities will also produce more nutrients for plants to grow and uptake more CO2 from the atmosphere. However, current assist models poorly simulate soil carbon dynamics, and there are huge uncertainties in model simulated soil carbon stocks. There's one big issue with these current models, which is that they do not explicitly represent the key mechanism controlling soil carbon dynamics, which is the interactions between microbes and the mineral surface. Indeed, most of the models represent soil biogeochemical cycling in an implicit way. So this whole below ground interactions between microbes, mineral surface, and plants remain a black box. So there's an urgent need to develop this micro-mineral explicit model and implement them in the global net model to better understand and predict the global soil carbon response to warming. However, we don't have good understanding of this diversities and trees of the microbial communities and their environmental control. For example, the nutrients, soil temperature, moisture, and substrate availabilities. So we have set up these two whole soil warming experiment to fill the knowledge gaps and help us develop the model. With this field experiment, we can address some non-standing questions. For example, how mineral adsorption impact microbial activities on the different warming scenarios, on how microbial biomass and trees change with soil depth and how they respond to climate warming differently. So with this enhanced understanding, we can develop explicit micro-mineral model and use them to better understand and represent the interactions, and then better predict the soil carbon response to different warming scenarios. Thank you.
Thank you very much, Mike and Jing Dao. Cheers. Xie. Who wants to play some more trivia? Come on, I know you want to. So, were you paying attention again? 4 SFAs supported by the Office of Biological and Environmental Research of the U.S. Department of Energy Office of Science will be moving into the BioEPIC building upon completion. What does the three-letter acronym SFA stand for? Is it San Francisco Authority? Is it Science of Fungi and Algae? Is it Scientific Focus Area? Or is it Science Forever Association? Your answers, please.
I have a personal favorite, I have to say.
Do tell.
Well, it's the Science Forever Association, clearly.
Clearly, we'll see about altering that.
If
but
it's
the real
correct, I'm not so
answer-
sure about this. Let's see.
Well, do we know what the real
Couple
answer
seconds.
is? I have a sense. Oh. What do you know? People are paying attention again, but they did like that Science Forever Association. I mean,
So did I
forever.
for sure.
Indeed. Let's move on.
So to introduce MCAFES, we have Trent Northern, and among the many hats Trent wears, he serves as Senior Scientist and Deputy Director of the Environmental, Genomic, and Systems Biology Division. He's also the Director of High Throughput Biochemistry of the Joint BioEnergy Institute, go JB, and Metabolomics Program Lead at JGI. When he isn't immersed in leading multiple scientific initiatives, Trent has led multiple successful Bay Area food bank drives. I'm sure you've heard about these. And in addition, he is an avid winemaker and grower of orchids. But relevant to today's event, Trent serves as the Laboratory Research Manager for MCAFES, where they are trying to understand and control micro-plant root interactions to support sustainable bioenergy. Take it away, Trent.
All right. Well, it's my pleasure to be here today at this exciting event. And so I'm going to be talking about MCAFES, yet another acronym for you. In this project, we're focused on the rhizosphere, which is where plant roots interact with soils. And this is an absolutely critical contributor to the global carbon cycle, like we just heard about. Plants release up to about 20% of their carbon through their roots as photosynthates. And why do they do this? Well, one reason for this is to attract and maintain beneficial microbes that naturally provide the plants with valuable nutrients. And so this makes it very relevant to sustainable agriculture. Now, these microbes, there's many challenges in studying these microbiomes. First of all, they're very complicated communities interacting with the plants. And most of these microbes have never been cultured. So we don't know much about their activities, the functions of their genes, and even less about their interactions. So MCAFES is developing a variety of powerful tools and applying them to explore the rhizosphere. So we're performing in-depth genetic and biochemical characterization of the different organisms, including fungi, which is on my Zoom background here. We're constructing spatial temporal models that allow us to simulate the movement and dynamics of these microbial communities. And this allows us to design experiments for using some of our really powerful new technologies, including in situ community editing using phages and CRISPR-Cas9 technologies. And why that's so powerful is it allows us to target a specific gene within a specific microbe within the community directly. So we don't have to isolate the organisms, and yet we can test our models and our predictions for the functions for those genes. And so by doing this, we can gain really important new biological insights into the mechanisms and interactions of these complex communities living in the rhizosphere. Now, we don't want to go use these out in the wild, in the natural environment. So it's really critical that we have fabricated laboratory ecosystems that are contained and controlled that allow us to deploy these advanced technologies. And that's why we've been developing some of these ecos that you've been hearing about. Fabricated ecosystems, which we call these ecofabs, which are little single plant size devices, and then larger meter scale contained systems, which we call ecopods. And these will be going into the BioEPIC building. And so we're really excited to collaborate with these other projects that you've heard about that are so complementary to our MCAFES project and really gain a lot of insights into the functions, interactions of organisms, and how they impact global carbon cycling. So with that, let me stop, and I'm going to introduce our first speaker, which is Kat Adams. Now, Kat is just about going on to her second year in the GLASS lab at UC Berkeley as a postdoctoral researcher, and she's going to be telling us about funky fungal farts, and I can't wait to hear more about that. So take it away, Kat.
All right, so I'll wait till I see myself big. Oh, okay, seems great. All right, so just like us humans, plants have a microbiome. So that means that microscopic organisms or microbes live on and in their leaves, stems, and roots. And these microbiomes are made up of mostly bacteria, viruses, and my favorite microbes, fungi. So the fungus that we study lives on plant roots and trades nutrients with the plant in a mutually beneficial symbiosis. Now, if you've ever smelled a plant, you know that they can produce remarkably strong scents like rosemary or garlic. So plants can fart. And by fart, I mean they produce volatile organic compounds that can float in the air. And it was known that our fungus can produce volatiles that impact the plant host, but I wanted to know if fungal flatulence can affect friendly bacteria that live on plant roots. So to do this, we designed a microbial thunderdome, an arena in which the fungal farts can reach the bacteria without the two microbes actually touching. So I would take one adorable tiny petri dish and put bacteria on it, another adorable tiny petri dish, put fungus on it, put them together in a standard petri dish, wrap it a bunch of times with science grade saran wrap to trap the farts. And the first time I did this, it was clear that fungal farts could actually inhibit bacterial growth. So up here, we have a control plate with no fungus, and you can see the bacteria are growing. Whereas over here, where there's fungus, there's hardly any bacteria growing. So I was like, great, this is amazing. On to the next thing. So to determine which genes are important for dealing with fungal farts, we did something called random barcode transposon site sequencing. And you don't need to know what it is, but just one of our key findings, which was, for one bacteria known as smelly, the effect of the fungal farts looked a lot like other experiments other folks had did where they acidified the bacterial food. And so we asked, are fungal farts changing the acidity of the bacterial food? And so to test this, we used these bespoke red petri dishes. And I made a bunch of different petri dishes with bacteria food that had different levels of pH, a measurement of acidity. We melted the contents of the petri dishes and ran the control samples through this machine that shines light through the samples, just to establish a baseline that we could then compare the experimental plates against. And then we ran the experiment. So on the vertical axis here, oh no, this way, we have pH. On the horizontal axis, we have days since plated. And in green, we have smelly growing by itself, actually lowering the acidity over time. Blue is a control plate to show that the food gets slightly more acidic over time on its own. But in red and orange, we have the fungal fart plates, the effect of the fungal farts on the bacteria food. And we can see that the bacterial food definitely turns more acidic over time because of farts. So in summary, just like us, fungi can fart, and those farts can impact friendly neighborhood bacteria that live in the plant microbiome. Those farts can even acidify the environment, which might help bacteria colonize plant roots. All right, thanks for listening.
Wonderful talk, Kale. Next, we have Tom Hessler, who's going to be, who's in the Banfield lab at UC Berkeley. He's in his second year, and he's going to be telling us about bacteriophage's contribution to the microbial community editing toolbox. All right, take it away, Tom.
Thanks, Trent. So we know that microorganisms and microbial communities interact with each other, and actually they interact a lot. They talk to each other, they fight with each other, and they trade and help each other out. But testing these interactions in the lab can be quite difficult and often requires taking these communities apart piece by piece and trying to put them back together again. But last year, members of our team demonstrated that it was possible to edit the genome of a specific organism, not in isolation, but within a complicated microbial community. And this meant we no longer needed to take these communities apart, and instead we could directly tinker with the target organism directly from its environment. Now, often the easiest way to test an interaction between two bacteria is to disrupt that interaction. For example, we might predict that one organism may be receiving a vitamin from another organism, And so we may ask the question, what happens if we knock out this pathway that produces this vitamin and see what happens to the rest of the community? Editing an organism's genome in this way actually makes it weaker relative to itself, which wasn't edited. And unfortunately, being able to make this edit in 100% of the cells is near impossible. So at this point, there's not much stopping the unedited cells, which are inherently stronger, from quickly outcompeting any organism which we've edited. So this is where we arrive at the problem. When we're testing these sorts of interactions, we need to be able to remove the fraction of organisms, of target organisms, whose genome we didn't manage to edit. Our approach to doing this is to use the highly specific and highly lethal solution of bacterial viruses, or bacteriophage. Bacteriophage have evolved to be really good at infecting and killing their hosts and producing more of themselves to infect other cells. But once we have these phage, we have to find a way to prevent them from killing the cells that we did manage to edit. So our approach to this is to take pieces of DNA sequence that match our phage and use them as a phage defense mechanism by incorporating these into CRISPR arrays, which we integrate into our target bacterium whenever we do a DNA editing experiment. By doing this, we leave the unedited cells vulnerable to infection, and we immunize any cell that received an edit will now survive the infection. And by using these methods, we're hoping to greatly increase the number of available interactions that we can study in the lab and use these to understand the role of microorganisms in the environment. Thank you.
Another great talk. Thank you so much, Tom.
Definitely super thought-provoking. Like, if a fungus farts in the forest and you're not there to smell it, does it have a fragrance? Just one of life's great mysteries, I guess. But thank you, Tom and Kat. And shall we move on? And Owen is with us, I believe. So last but not least, we have our fourth scientific focus area, watershed function, led by Owen Brody. Owen hails from Ireland and has made Berkeley Lab his home for a long half-life. He is the director of the Earth and Environmental Sciences Area, Climate and Ecosystem Sciences Division, and serves as associate adjunct professor in the Department of Environmental Science, Policy, and Management at UC Berkeley. After a long day of advancing Earth environmental science, he works out on the rugby pitch where he can dish it out as well as he can take it. Owen is also an avid cyclist and is preparing for the Tour de France, at least preparing to watch it. We'll hear now about the watershed function, SFA. Owen, take it away.
Great. Thanks a lot, David and Lydia, and also to Lydia and Christina and everybody involved in preparing for today. So I know a huge amount of work went into this. Also, thanks for the unexpected promotions. I'm the deputy director of the division. I also say I co-lead the SFA with Ken Williams and Michelle Newcomer, but I'm really delighted to be here today to represent the watershed function SFA. You can see behind me, I think if I move out of the way, that's the East River watershed. And that's our field observatory. It's a beautiful location to do science. It's in Crested Butte in Colorado. And this is the headwaters of the Colorado River. The Colorado River is extremely important for the Western US, both for the communities that live along the river, but also for agriculture, for energy production, and for industry as well. We've made this our research home. And thanks to great work by Ken and others, have turned this into a community watershed. And our scientists from Berkeley Lab and also 50 other institutions are now researching how climate change, that's droughts, floods, wildfire, etc., are impacting the functioning of this watershed. And we literally go from the bedrock to the top of the forest canopy and are working from microbes to mountains. And that's a national lab scale challenge, and actually beyond the national lab scale challenge. So you can imagine, to build a predictive understanding of how an ecosystem this complex works, It means, number one, working across disciplines, but also working across the range of length scales that you can see to my right over here, and working between not just the field system, but also the lab and the type of model ecosystems that Trenton mentioned. What's really important, though, is to connect knowledge across these scales. We have to use approaches that connect the scales. And so a big approach for us is using remote sensing and ground-based sensing across various scales, machine learning to make sense of that high density information, and then advanced computer simulations, process models to bring this all together. So for me, that's exactly what BioEPIC is all about, bringing disciplines, scales together, wrapped around a common approach to data analysis, data synthesis, and in an open and sharing environment. And we're really excited to get going. I think we've already started interactions across the different teams that you see here today, and to be able to bridge these scales with our friends in biosciences across the lab and beyond as well. So this afternoon, I'm thrilled to introduce you to two amazing scientists on our Watershed team that really do connect these scales to from molecules to ecosystems. So first up, we're going to hear from Dr. Hannah Naughton. Hannah is a postdoc who's been with us for about 1.5 years, and Hannah is going to present on mountains, microbes, and microsites. Oh my indeed. Take it away, Hannah.
Thanks, Owen. Let's see, I don't know if people can see my screen yet. I guess we just stop share. Oh, okay, people can see me. Great. So I study soil microbes, which host key ecosystem services, such as the provision of nutrients to plants like nitrogen, and also the breaking down of organic matter to small pieces that can stick on soils and be stored long-term. However, we struggle to model how microbial activities impact ecosystem-wide processes. And I argue this is because of the complexity of the soils and the microbial habitats in them. 1 gram of soil, about a teaspoon, has about 1 billion microbial cells. And it also ideally has up to 50% empty pore spaces that can range in volume or diameter, I should say, from 1 millimeter down to the width of 1/100th of a piece of copy paper. And we often assume that these pore spaces are completely aerobic, meaning oxygen is readily available to microbes to consume the carbon in the soil and return it to the atmosphere as carbon dioxide. In reality, many soils actually have a lot of anaerobic pore spaces due to water or high carbon environments, where the lack of oxygen slows down the microbes churning out carbon dioxide by up to 100 times. And it might also help the microbes who produce more potent greenhouse gases like methane thrive. So the question is, how do we characterize these microbial habitats that vary in the scale of millimeters across a watershed or larger landscape? We need proxies, because to go out, collect soils, haul them to lab, preserve them, and then get computed tomography scans like this that are so computationally expensive, I couldn't even open it on my laptop, is not going to work. Plants are a great option because, as you can see, we can see different plant communities and remotely sense them down to meter scales, as Nicola will discuss, hopefully. And moreover, the plant roots create the soil aggregates that host these microbial habitats. Topography or mountains are also a great option for remotely sensing microbial habitats, because the slope aspect towards the sun and also relative height in a landscape determine the movement of water, carbon, and small particles, which drive the availability of oxygen to microbes. I go to this lovely watershed site and collect soils from under different plant types and different positions on this hill slope, take them to lab, and look for the redox microsite habitats of microbes. And I also characterize what the microbes are doing in these habitats. Are they quickly removing nitrogen? Are they going to rapidly produce carbon dioxide based on sequencing their genomes and also looking at their volatile organic compounds produced, so what they're breathing? We hope to statistically tie together what the microbes are doing in their tiny sites and tiny aggregates to landscape meter scale variations in plants and topography, so that we can predict going to the future how the microbes in these watersheds will respond to environmental perturbations like decreased snowpack and increased rainfall. Thank you.
Right, thanks, Hannah. That was awesome. Really super. So next we're going to hear from Nicola Falco, and you can see in my slide behind me here, Nicola is moving up in scale. Nicola's been with us for just over 5 years and is a great success story. Began as a postdoc, and is now a research scientist. And Nicola is going to talk about sensing the invisible, connecting the hidden structure of watershed ecosystems from space. Take it away, Nicola.
Thank you, Owen. So what if someone told you they want to observe the impact of microbe in a small area of soil from space? It seems far away to study something smaller than a human cell, but this is exactly what we did, and we used remote sensing and machine learning to do it. We work on a mountainous watersheds, which are considered the water tower of the world, as they provide 60 to 90% of the water resource worldwide. Climate change is impacting these regions, while declining snowpack, exceptional droughts, and intense fire season. This has a dramatic effect on ecosystem functioning, affecting the interaction between plants, soil, and microbes. Now, let's say you wanted to understand the scale of a drought impact in a region as vast as the Rocky Mountains. We will need to pinpoint how plants become impacted by changing water dynamics around them. Scientists have been studying this, but mainly at plot scale. The trouble with that is the watershed contain incredible landscape diversity, where things like temperature, elevation, topography, and sunlight can be dramatically different from meter to meter. So the question is, how can we use the plot scale insights from scientists such as Hannah to understand microbe, soil, and plant interaction at watershed scale while capturing the full heterogeneity of this landscape? This is where remote sensing and machine learning can be powerful tools to scale up the characterization of complex ecosystems. Remote sensing refers to sensors mounted on satellites or airplanes that allow us to gather data where no one else has, so we can tell new stories about how these watersheds are changing. In my research, I use particular sensors based on spectroscopy to capture hundreds of colors. With these sensors, I can identify even slight gradient differences in the color of leaves. So I can understand the species of plants and create maps like the one here on my side, understand the traits of these plants and even their overall health at scale. Then I use machine learning to integrate this data with ground observation to derive relationships between plants and surrounding landscape. At the plot scale, researchers are looking at the impact of microbes on the plant. With remote sensing, we do it backwards. We look at the plant activity and we infer what that may mean about microbial activity. We use plant and topographic information from remote sensing as a proxy to understand what microbes may be doing below the surface. A microbe is 1/1,000,000th of a meter, and the watershed we study is about 300 square kilometers. Remote sensing is a tool that has the power to link these two vastly different scales together to arrive at novel insights. See, we can study microbes from space.
Outstanding. Thank you, Nicola.
Yay, Hannah and Nicola, thank you very much indeed. Grazie mille. Lydia, now what?
Now the time has finally come and it's your chance to vote for your favorites. I encourage all of the presenters to join us and turn on the camera. Let's see some faces, guys. And audience, let's also show your appreciation and share your words of encouragement in the chat now. So with that, I think the link for voting has now commenced. It should be this in the slide that's going to be shared soon. Yes, there we go. And you will only have 1 minute to cast your vote. So don't hesitate and vote now. We're doing a quick recap during this minute. And we have heard from ENIGMA, represented by Fang Chao Song and Audrey Leeman Wang. We have heard Belowground biogeochemistry from Mike Rowley and Ching Tao. We have seen MCAFES being represented by Kat Adams and Tom Hessler. And last but not least, Watershed Function with Hannah Naughton and Nicola Falco. So
Okay,
choose
audience,
your favorites.
Please. Yes, you only have 5 more seconds to log your vote. That's 4, 3, 2, 1. Voting is closed. Now, I have the privilege to introduce you to the Berkeley Lab leaders who will announce the winners. I present Associate Laboratory Director for Biosciences, Paul Adams, and ALD for Earth and Environmental Sciences, Jens Berkholzer. Jens, please take it away.
Thank you so much. You guys hear me, right? Good, okay. It was really great to see the research that will be done in the newest building on campus described in these fascinating presentations. Congratulations to each of the participants for their engaging talks. I really enjoyed each one of them. It's very hard to pick a winner here. With this level of talent evident today, I'm eager to see how the BioEPIC facility will bring together under one roof our two areas' incredible staff and new capabilities, thereby further advancing an already close and fruitful collaboration between biosciences and earth and environment sciences at Berkeley Lab. Over to Paul.
Thanks, Jens. Many thanks to all of you who presented our science focus areas today. Great, fantastic presentations. And after so many years of planning, it's been a while. It's fantastic to see the building actually rising up from the ground. It's remarkable how fast it's going up. And to think we're just a couple of years away from planning our move in. And the BioEPIC building is going to provide unique capabilities you've heard about to understand microbe and plant function in both fabricated and natural ecosystems. Now, critical for success are the researchers who will be performing their work in the building and linking that work to studies in the field. And I think you can all agree after today's presentations, you can see that we're in really safe hands. And I look forward to seeing many of you in the new building in the not too distant future. So we should get on with the awards. Jens.
Right. If I wish we knew who the awardees are. Will I get a text here or a
golden envelope?
The winner is. Okay, here we go. So without further delay, let's announce the winner of our Honorable Mention Award, who will get a spot award for $100. The votes are in, and the honorable mention goes to Nicola Falco, research scientist in the Watershed SFA, and he presented Sensing the Invisible and so forth and so forth. Congratulations, Nicola.
Thank you, everyone. I'm glad that I reached the audience.
Awesome. Paul?
Great. Yes, thanks. So next we have the first runner-up who will receive a spot award for $175. And this award goes to Hannah Naughton, postdoctoral researcher in the Watershed SFA, who spoke about mountains, microbes, and microsites. So well done, Hannah.
Thanks so much. Glad you all could join. And congrats, BioEPIC.
Awesome, awesome. So now, last but not least, our first ever BioEPIC Research Slam champion, who will get a spot award for $250 and bragging rights, of course. And this is Kat Adams.
Yay.
And she's a postdoc, and we heard about the funky fungal forest, which was awesome.
Oh, thank you, everybody.
Wow, congratulations again to our BioEPIC champ, Kat, and of course, all the participants. It is an honor to welcome all of you guys to the growing family of slammers. And before our slamming time together ends, we want to invite you all to join us for the DOE BioEPIC Earth Day event on Friday, April 22nd at 3:30 p.m. Pacific time, with the theme of investing in our planet. This will be a collective celebration of energy and environmental research across the National Lab Complex, while demonstrating the commitment, excitement, and importance of STEM with the local and national communities.
The Berkeley Lab session will celebrate BioEPIC and will include an overview by Owen Brody, the Smart Soil Testbed by Yuxin Wu, and EcoFabs and EcoPods by Trent Northern. The session will wrap up with a conversation between Trey and Rachel Jang, a high school student. Oh, Trent and Rachel Jang, a high school student from San Francisco, who was in the Berkeley Labs apprenticeship program last year and used the EcoFabs to learn about plant processes. And with that, I say thank you, Lydia. Thank you. You'll always be a champion in the pantheon of Berkeley Labs Slam champions. We thank our worldwide audience for tuning in today for the BioEPIC Slam. Follow upcoming events at #BioEPIC and be well. Hope to see you soon.